Okay, so I'm Dan Fordham. In the interest of everybody's life, I'm going to try and keep this as brief and enjoyable as possible. I'd like to thank everyone for giving me the opportunity to speak today. And we're going to talk about nanopore sequencing and how we consider it to be, you could consider it to be live and direct. So one of the things about nanopore sequencing, and something that we talk about quite a lot, is its portability and the low barrier to entry for people to be able to use. So what this has particular applications to is things like an outbreak situation. Now, if we ignore COVID, which we could consider the elephant in the room for the moment, there have been many examples of people deploying MinION sequencers worldwide to areas that are low income or lower resourced, and tracking and monitoring different outbreaks and as they occur and in situ. Now, there is an ongoing and increasingly concerning swine fever outbreak that's happening worldwide. But in the past, the Ebola outbreak in Guinea in 2015, 2016, and the Zika outbreak in Brazil that's still going on in some form is of particular interest, and people have been deploying nanopore sequencing in those areas and being able to make decisions according to the outcomes of their sequencing. So here we have again the picture which I highlighted already, where in 2016 the Arctic Network identified that there was an ongoing Ebola outbreak and packed off Josh Quick with a lab in a suitcase to sequence Ebola genomes as just after the majority of the outbreak had happened. But from that, they were able to gain some great insights in how the outbreak occurred and how it was transmitted, and the lessons from this they then passed on to the Zika outbreak in Brazil, and they were going around in a van out there. So why use nanopore sequencing for this? Well, rapidly evolving outbreaks can present many complex challenges, and being able to deploy a sequencer or a sequencing network allows people to implement rapid testing close to the sample of the outbreak. This in turn can facilitate the tracing of individuals, and then it enables in a larger format a decentralized model for rapid testing. Obviously, not obviously, but for those that are aware, nanopore sequencing delivers the answer very quickly, in some cases real time, and this enables people to implement scalable solutions for in-field rapid testing. And if you're interested in that and you want to move to a more centralized model, then larger batch sequencing. Now, I've sort of gone around this circle already. I seem to be a slide ahead of where I wanted to be, but you can consider with this, what we're doing is how genomic data can inform your disease control. So there is the ability to identify an outbreak at the very beginning, or early on, and distinguish it from the other pathogens that might be involved. It enables people to analyze the genome, get the variants, and that helps to inform the design of diagnostics, the therapeutics, and vaccines very early on. It helps people to understand transmission, and if you're able to sequence a number of individuals very quickly, and you can start to see what the patterns of transmission are, and this helps us to monitor the outbreak, start to monitor the impact of any interventions that are going on as well. And this then will lead us to provide more precise and deeper information about how to manage that outbreak, how to prevent outbreaks, and how to design interventions in the future. Now, nanopore sequencing, one of the benefits, or one of the true things about it, is that it's the same technology on all of our devices. And so from that, if you want more sequencing, you can just add more ports. So we can move through our family of sequencers from the very smallest, which is the Flongle or the MinION, to a Mark 1C, which is, I don't know if you can see my pointer here, which is a MinION with genomics with base calling capacity on top. A GridION, which is effectively five MinIONs on top of a supercomputer to be able to deliver 5 times the throughput you get from a single MinION. And then the PromethION, which is either 24 or 48 formats, where each individual flow cell can be run on its own, and it can be run in a batching format. It can be run with debatched. It can be run however you want it to be. So it enables people to be able to be more flexible with their sampling. So you can start to design your experiments, or not design your experiments, and just be able to react as things occur. Now, we like our sequences to be accessible, so they are low capital cost and very simple workflows. They're fast, as mentioned before. They're increasingly accurate. They're flexible. So this is going back to being able to do, not having to wait for a number of samples to accrue before you start a run, and just running as you want. Or if you want to get more more higher number of multiplexing on your devices, then we have ways to get to that as well. So when we say things are scalable, it means you could run 1 sample a day if you wanted to, or up to 5,000 samples a day. And it's completely up to the user what they want to do about that. And all these things tied together mean that it's deployable, and these portable devices are available very rapidly. So if a new outbreak were to occur, and we know it may, then a MinION can be available the next day, and a GridION within a week. And this enables us to enables us and the users to flex depending on different outbreaks and different situations and different capacities and so on. So in late 2019, a novel disease was identified in Wuhan in China. I'm not sure where it come from. And using a combination of nanopore and short-read sequencing data, they were able to characterize the SARS-CoV-2 genome very early on. In January in 2020, one of the last airplanes to leave and land in Wuhan was carrying 200 MinION sequences. And they were shipped to China to support the Chinese CDC and increase their sequencing capacity in order to try and track this outbreak of this novel coronavirus. So this is a real example of rapid deployment of nanopore sequencing to characterize the SARS-CoV-2 outbreak in China. And of that, there were 60 kilos worth of equipment, and there were 75 labs that were then able to form a network to monitor this outbreak. So the next question is, well, why use Nanopore for doing this? And as sort of alluded to before, we were able to rapidly sequence SARS-CoV-2, and that helped people to understand the transmission, to investigate the clusters, and identify the new variants and monitor their prevalence and so on during the outbreak, hopefully that we're coming to the end of soon. People were able to get started quickly, and are still able to get started quickly with simple end-to-end workflows with little prior experience needed. I will say that we probably will need at least a passing introduction to a pipette, and there is a short waiting time and a plug and play setup. And this is available to people from as little as $1,000 or to £1,000. And depending on what any user wants to do, they're able to either sequence those small amounts or have a highly multiplex, high throughput system that is able to sequence thousands of samples on either a GridION or a PromethION with no capital costs. So the Arctic Network, a number of years ago, were interested in the possibility of, say, a pathogen outbreak in the future. So they were well placed to be able to help with the tracking of the COVID pandemic. So they had already developed a rapid and effective method of library prep that would, as mentioned before, had been deployed for Ebola and Zika sequencing, and with very quick turnaround, they were able to apply this to SARS-CoV-2 sequencing. And one of the driving forces of the Arctic Network is to deliver a real-time approach to provide rapid, actionable epidemiological insights, to reconstruct the epidemiological processes that drive an outbreak by sequencing the entire viral genomes as quickly as possible, and to monitor rapidly evolving viral genomes to accurately record or monitor the drift of the virus. So how did they do this? Well, to begin with, what they were doing is to have 400 nucleotide amplicons with 100 nucleotide overlap, so a PCR tiling approach. And for Ebola, they had 38 amplicons. For Zika, they had 35 amplicons, and these were able to capture the entirety of the virus very easily. So they'd also gone as far as providing a bioinformatic answer to the data that had been produced from these things, and that's freely available on GitHub. It's very comprehensive. It's called Rampart, and they have a strong SOP that is provided alongside it as well. So Nanopore Sequencing was able to deliver full strain ID and transmission mapping, in collaboration with the Outbreak network, and allow the detection of new variants from Nanopore Sequencing. The rapid whole genome was able to then easily identify the SNPs to assign the variants, and again, the cost per sample can be as low as $9.55 if you're driven by that. So we can quickly go over the timeline and how we worked in collaboration with them, and how the rapid deployability of Nanopore, the rapid change of Nanopore, of Oxford Nanopore in general, was able to work throughout the pandemic to really start to deliver some of the genomes that we sequenced on for SARS-CoV-2. Let's skip this slide. So and this slide. So here's another example of how a country, for example, was able to deploy Nanopore sequencing very quickly in a distributed manner. And so in Colombia, they identified that they were obviously they were affected by the COVID-19 pandemic, and so they strategically placed a number of MinIONs throughout Colombia, and also larger scale sequencing centers in Medellin and Bogota as well, where they're able to work in a more centralized manner, being able to produce more data where it's needed. They've also thought beyond that, and so post-pandemic, they were starting to think, well, once we have this this network in place, what are we going to do with it? So they've thought about, well, actually, we don't necessarily just need to sequence the virus with these. We can also look at biodiversity. We can look at emerging viruses and disease surveillance, which would forefront in most people's minds. TB and mycobacteria, clinical biology, and then there's RNA-seq for cancer tumors, and AMR and epidemiology, and all the things that are completely possible with any one of the Nanopore devices. So one thing that is of interest to people going forward is being able to monitor an outbreak and monitor other diseases or pathogens in a less invasive manner. And this group in Nice were able to monitor the SARS-CoV-2 in wastewater samples that were collected monthly anyway from 21 locations. And what they're able to do is actually say, well, 2 weeks before an outbreak actually kicks off, or a variant outbreak is really monitored through people self-identifying, you're able to pick it up in the water. And so they were able to extract the RNA from the wastewater, and were then sequencing them and being able and ready to start to deploy interventions when they're needed and in the future. So as said before, Nanopore sequencing is scalable. You can sequence 96 SARS-CoV-2 genomes in a day if you want to. On a gridiron, you can do 480. On the PromethION, you can do greater than 1,000 SARS-CoV-2 genomes in a day, and that's really limited by manpower, logistics, and so on. So what is Nanopore's contribution to this, to the sad state of the pandemic that we're still in? Is that 400,000 Nanopore generated genomes are available and uploaded to GISAID. This has produced over 200 publications, and the particular interest to me, and I think should be to everyone, is that these have come from 70 different countries as well. And the low barrier to entry and the low cost of sequencing, and all the things that Nanopore talk about democratizing sequencing, is borne out through all these different people from different countries being able to monitor what's going on in there. And so thank you very much.